My name is Ryan Christian, and I'm the Technical Director at Yakima Chief Ranches. I oversee technical improvements at YCR and manage the YCR Research Laboratory.
My name is Josh Peralt, and I am the Production Lab Manager at Yakima Chief Ranches. I make sure the production side of the lab is running smoothly and hitting its targets in terms of propagation.
Part of the mission of our lab is to produce clean, virus-free hop starts and ensure quality control throughout the propagation supply chain. This is the plant tissue culture grow room at Yakima Chief Ranches, and this is kind of where all the magic starts for our clean plant supply chain. This room is at a balmy 80 degrees and is about 50 to 60% relative humidity. The process of micropropagation starts with induction of small pieces of field-grown or potted plants. Here, a virus-tested plant is cut into individual pieces, surface sterilized, and grown in individual test tubes. The best single plant to pass through multiple cycles becomes the mother plant to all cuttings in that propagation year.
During micropropagation, lab technicians break down the plant into individual nodes and replant them into fresh media.
A node is the point at which a pair of leaves meets the stem, and each node has many dormant buds which will regrow given the right hormonal signals. So within a single month, plants produce 5 to 10 new nodes that can be replanted to multiply the culture, and in so doing, we can start with a single plant in the early spring and produce tens of thousands every week by fall. So this is an example of plants in the initial phases of tissue culture. These were newly brought in from the field, sterilized, and then brought into individual tubes. These will stay in here for about 10 days, allow them to grow new shoots, and then we'll transfer it to new media and start the propagation process.
To keep conditions sterile and free of contaminants that could affect the health of the plants or our workers, our micropropagation technicians work in portable cleanroom spaces called laminar flow hoods. Tools are sterilized between each and every vessel using a high-temperature glass bead sterilizer. We use a jelly-like material called agar in our media, which is similar to the material used in Petri plates. The agar contains all the water, nutrients, and vitamins that the plants need to grow well without needing strong roots.
This is an example of a newly propagated plant just born yesterday. So these are HBC 394 or Citra plants. And this is the same variety at another stage in the process.
It is essential that cultures remain clean so we are not propagating dirty and contaminated materials. Technicians check the seed cultures at least 3 times throughout each cycle, and once more right before they open the vessels again. Plants destined for the greenhouse are checked for vigor and for the amount of rooting, which greatly affects how quickly they will transition into the greenhouse. Batches are genotyped before propagation to ensure that they are true to type and periodically tested for virus contamination. Propagating plants in this way allows YCR to move quickly to respond to farmers' needs, whether that is replanting with virus-free stock or scaling up new and improved varieties. YCR's tissue culture program is designed to provide an excess of very clean propagation stock that greatly decreases the risk of reinfection by viruses and viroids between the lab and field.